all participants are in a listen-only mode until the question and answer session of today's conference. At that time, you may press star 1, unmute your phone, and record your name to ask a question. I would like to inform all parties that today's conference is being recorded. If you have any objections, you may disconnect at this time. I would now like to turn the conference over to your host, Ms. Laura Bleacher. Thank you. You may begin.
Good afternoon. I'm Laura Bleacher from NASA's Office of Communications. We have an incredible panel of experts that are ready to answer your questions about the research investigations that they're getting ready to launch aboard SpaceX's 28th Commercial Resupply Services mission for NASA to the International Space Station. NASA and SpaceX are targeting no earlier than Saturday, June 3rd, 2023 at 12:35 p.m. Eastern Time for the launch of the company's Dragon spacecraft atop a Falcon 9 rocket from Launch Complex 39A at NASA's Kennedy Space Center in Florida. Here to talk with us about some of the research launching aboard the SpaceX Dragon are Dr. Kurt Costello, Chief Scientist for the International Space Station Program, Dr. Olivier Chanrion, Principal Investigator for Thor Davis, Scott Copeland and Dr. Ali Huang, who will speak on Genes in Space 10, Tony Pellerin, who will discuss the Canadian Space Agency's CubeSat, and Dr. Annalisa Paul, Principal Investigator for Plant Habitat 03. All of our guests will give a quick overview of their research, and then we will open the lines to questions. We encourage you to ask questions to the scientists about their experiments. If we have time at the very end, we'll take additional questions for any of our speakers. Your phones are on mute now. To get into the question queue, please press star 1 at any time. We'll call on you, and the operator will open your mic so you can ask your question. Please stick to one question at a time, and then re-enter the queue with a second question if you would like. We are also taking questions using social media with the hashtag AskNASA. About 1 hour after we conclude, you can listen to a replay of this teleconference by dialing toll-free 888-566-0411 or by visiting the NASA Videos YouTube channel. Again, that's 888-566-0411. To start off, we will hear from the International Space Station Program Chief Scientist, Dr. Kurt Costello, who provides leadership and support for science performed on the space station. Over to you, Kurt.
Thank you, and good afternoon, everyone. Today on the International Space Station, we've had a successful undocking of the Axiom 2 SpaceX Dragon vehicle. Now we're eagerly awaiting the arrival of SpaceX's 28th commercial resupply mission. The International Space Station is an amazing multidisciplinary lab offering unique access to long-duration microgravity and the low Earth orbit space environment. Each CRS mission brings a variety of new research to the station. This mission has some exciting new technology demonstrations and brings the hardware and supplies needed to complete the most ambitious plant research we've conducted to date on ISS. This SpaceX mission also brings 2 more iROSA solar array wings to augment the ISS power generation capability and enable the next decade of science on board. The research highlighted today will add to the over 3,700 investigations already conducted on the ISS and focus on technology demonstrations for NASA and our international partners. Several of our researchers will be joining us today with details on their research. And now I'll hand it over to Olivier.
Thank you, Kurt.
Yeah,
Yep, thank you. Now we will hear from Dr. Olivier Chanrion, Principal Investigator for the European Space Agency's Thor Davis investigation. He received his PhD in Applied Mathematics in 2002 and is now a Senior Scientist at the Technical Institute of Denmark. Olivier, over to you.
Yes, thank you, Destiny. I represent the Thor Davis mission experiment, and the scientific purpose of the experiment is to conduct video camera observation of thunderstorms and their electrical activity. We want to take advantage of the vantage point of the International Space Station, since from there we can look at thunderstorms from above. In fact, with this investigation, we want to focus on the activity at the top of the clouds and above. Scientifically, we aim at understanding better the role of thunderstorms on the Earth's atmosphere and climate by looking in particular at their impact at stratospheric altitude. With this Thor Davis experiment, we have also technical purpose because we want to observe the thunderstorm with a new kind of camera, a neuromorphic camera, that will allow us, in fact, to look at the thunderstorm activity at an equivalent of 100,000 frames per second. And yes, at side goal, we want to experiment with this kind of technology, this kind of camera, the neuromorphic camera.
Great. Thank you, Olivier. We will now take questions. Again, please press *1 to be entered into the queue.
Thank you. We will now begin the question and answer session. If you would like to ask a question, please press *1, unmute your phone, and record your name. If you need to withdraw your question, please press *2. Again, to ask a question, please press *1. And let's see.
I see we do have a question from Jim Siegel from nasatech.net.
Hi, everybody, and thank you for taking my question today. I live in Central Florida, and Central Florida is supposed to be typically known as the lightning and thunderstorm capital of the United States. And I wondered if you had any comment about whether when you're studying thunderstorms, if you're going to be focusing in on Central Florida, for example, or what you know about the thunderstorms in Central Florida, and are they different from thunderstorms perhaps in other parts of the world? Thank you.
Thanks. First, with this Thor Davis experiment, we'll take all the opportunity we have to see storms. If they are over, Europe or US or Asia. So, but I'm not sure we can really decide in advance how much storm we'll see over Florida. But I hope so somehow, because usually the storm over Florida are quite powerful, and they might contain a lot of convection. And when we want, so since they have a lot of convection, they can go really high in altitude. And since we want to focus on what happened at really high altitude, what the thunderstorm does at stratospheric altitude, the storm of Florida are so to say, quite interesting for us.
Thank you, Olivier. Next, we will hear from Scott Copeland and Dr. Ali Huang, who will speak to the Genes in Space 10 experiment. Scott is the Boeing Director for Research Integration on the International Space Station and a co-founder for the Boeing Genes in Space Molecular Biology STEM Education Competition. Dr. Ali Huang is the lead research scientist at Mini PCR Bio and student mentor. First, we will go over to Scott and then to Ali. Scott?
Thank you very much, Laura. We're excited to be here today. And the Genes in Space competition was co-founded by Boeing and Mini PCR Bio in 2015, and it challenges students in grades 7 through 12 to design an experiment that can solve a space exploration problem through DNA analysis using the unique environment of the International Space Station as a test bed. To date, we've received over 5,000 proposals from 9,800 students. And each year, the finalists are chosen from a competitive group of applicants from across the country. The proposed experiments have covered a wide range and variety of topics. The top 5 finalist teams then receive mentoring from MIT or Harvard University researchers to fine-tune their proposals in preparation for our final selection. Results of the Genes in Space cutting-edge research are routinely published in peer-reviewed scientific journals. And today we'll hear about last year's winning experiment that was selected from over 600 proposals that will fly this weekend. With that, I'll turn it over to Allie.
Yeah, thanks, Scott. So I worked closely with our last year's winner, Pristine, to develop her experiment to be launched this weekend. And her experiment is actually going to be looking at a method for measuring the length of DNA fragments. That is a pretty important part of a lot of different biological applications, such as diagnostics or just understanding biological mechanisms. And these applications can be really relevant to space. For example, Pristine's project was actually inspired by telomeres. These are little caps, genetic structures that protect our chromosomes and shorten with age, but Pristine was inspired by the NASA twin study, which actually found that telomeres actually lengthen in space. Unfortunately, to understand this phenomenon and any other phenomena related to DNA length, the samples usually have to be sent back down to Earth from the International Space Station because there is no accurate way to measure DNA length in space due to the fact that the usual methods are really difficult to do in a microgravity setting. So her experiment that's going up this weekend will be a way that we're validating a means to measure and analyze the length of synthetic DNA on the ISS using fluorescence samples that can be visualized and analyzed by eye while on board. And we hope that these experimental findings can provide the baseline research needed for future research investigations from like Pristine's inspiration from astronaut telomere elongation to even things like molecular diagnostics aboard the ISS. So happy to take any questions now.
Thank you. We will now take questions for Scott and Allie. Again, if you are joining us online or on social media, you can use the hashtag AskNASA to ask questions as well. For Scott, how did the Genes in Space competition come about, and what sets this competition apart from others?
Okay, well, Boeing is continuously looking for emerging businesses that can take advantage of the unique environment of the ISS. We met our co-founders while participating in Boston at a new business accelerator program. Back in 2014, we realized there was a significant gap in the process, not in the process, but in the equipment that was available on board the ISS to perform molecular biology experiments. They had a unique device, which is basically a thermal cycler that was very small, compact, didn't take much power or resources, so it was a perfect fit. So we purchased a few of those items, and then we got them space qualified, and we partnered together to figure out how best to use it. And so at that point, we decided to turn the question over on how best to use it over to the students. So that was kind of the genesis of the Genes in Space program. I think really the two things that set this apart from other STEM education competitions that I've seen is the mentoring that takes place between the Harvard and MIT researchers. They donate their time. They're doing their own research, but they donate their time to help these young students to perfect their experiments. And the other item that I think sets it apart is the research that's coming out of that. Every experiment to date that we've submitted to journal articles from peer-reviewed articles has been accepted.
Thank you, Scott and Allie. Now we will hear from Tony Pellerin, Manager of Space Science and Technology for the Canadian Space Agency. Tony has been with the agency since 1994, involved in many spacecraft missions during that time. As technical lead, he has provided support to all CubeSat design teams for the Canadian Space Agency CubeSat projects. And I'll turn it over to Tony.
Thank you. So this is a very exciting moment for the Canadian Space Agency, but more importantly, a very exciting moment for several young Canadian students who worked very hard over the past 5 years to design and build their CubeSat that were part of the Canadian CubeSat project. On this launch, we will have 5 Canadian CubeSat that will be part of the NanoRack 26 deployment mission. We have RadSat from the University of Saskatchewan. We also have UQPIC from the Western University in Ontario, SC Odin from Concordia University in Quebec, and Essence from the York University in Ontario, and finally IRIS from the University of Manitoba. 2 of these missions, Essence and IRIS, carries novel science payload. So I'll explain very briefly what they are and what kind of science they will be conducting. So Essence from the York University is basically an Earth observation mission. It will observe the Arctic ice and the permafrost thaw in the northern portion of Canada using a camera with a fisheye lens. And it will also observe the forest status. It also carries a solar proton density measurement instrument. And finally, they will also conduct attitude control experiment using novel algorithm with the ADCS subsystem they have on board. Now, the second one, IRIS from the University of Manitoba. This one is pretty cool. It will carry 20 geological samples to study the effect of space environment on these samples. So basically what it is, they have an aperture opening on the side of the CubeSat, and they're exposing these 20 samples, which are basically the size of a penny, to the harsh environment of space, and they have camera on top of it that will take picture, and they will, in a short time, try to see if they can detect deterioration of those samples due to the harsh environment of space. So these missions will be deployed from the space station using the NanoRack system. We're expecting at the end of July. So in a nutshell, that's what it is, and I'm ready to take any question that you may have.
Thank you, Tony. We will now take questions. Again, please press *1 to be entered into the question queue. And I see we have a question from Jeff Faust of Space News.
Good afternoon. I'm curious, how did CSA select these 5 CubeSats, and were these CubeSats funded entirely by the agency, or did the universities also have to raise money to help develop these satellites?
Yeah, that's a very good question. So the intent of the Canadian CubeSat project was to offer opportunity across the country from coast to coast. So we've down selected a university per province and territories. So the Canadian CubeSat project had in total 15 missions. Now this is the third batch that we're going to launch. So they were selected based on the proposal that we've received on the scientific objective and the way they're going to train the student, because the goal number 1 of the project was to train students on how to design, but build and test CubeSat, and finally operate them in orbit. And in terms of budget, the CSA provided grant contribution to the team, and most of the team managed to secure some funding through their own funding searching. But CSA also took care of the cost for launching those CubeSat through the ISS.
Thank you, Tony. I see we also have a question from Jim Siegel.
Thank you for taking my call again. And I'm interested, what I like to do is try to write to the average citizen, in this case it would be the Canadian citizens, and explain to them what the value is to them potentially of many of the investigations that are aboard the ISS. So, for instance, these 5 investigations, would you care to speculate on what benefits the average citizen might get eventually from whatever is learned from these investigations? Thank you. Of course. So if we take Essence, for example, it's actually looking at the permafrost deterioration, for example. And this is a direct impact on the climate change. So right from the bat, we can say that this type of mission will bring great benefit to Canadian, but not only Canadian,
but the world population in making sure that we understand those climate change effect. Okay, so that's but also keep in mind that the objective number one was to train students on a real space mission. And eventually all those students, we had over 2,000 students participating in the CubeSat project in Canada. And they will migrate to the industry and working in the space industry and eventually work on mission that will also impact Canadian and the rest of the world life.
Okay, got it. Thank you.
Thank you. We will now move to Dr. Annalisa Paul, Principal Investigator for NASA's Plant Habitat 03. Annalisa is the Director of the Interdisciplinary Center for Biotechnology Research at the University of Florida and a professor in the Department of Horticultural Sciences. She and her colleague Rob Furl lead the university's Space Plants Laboratory, where they have been conducting research on the molecular responses of plants in space and other exploration environments for more than 20 years. Annalisa?
Thank you. Yeah, so my colleague Rob Furl and I are interested in how plants respond and adapt to the novel environment of space. And you got to remember, it's absolutely outside the evolutionary experience of any terrestrial organism. And so it's very interesting to see what kind of tools plants use to adjust. And so in one of our previous ISS experiments called Apex 04, we found that some of those tools that plants use to adjust are associated with what's called epigenetic change. So the objective of our current experiment, this PH03 experiment that's going to launch on Saturday, is to determine whether these tools that are developed by the first generation of spaceflight is going to be transferred to the next generation to help that generation better cope with the stresses of spaceflight. In other words, you know, if the parents grew up and set seed on the ISS with the next generation, those seeds that developed on the ISS, would they be better able to deal with the spaceflight-associated stress that we see plants mount in that environment? We do see this phenomenon on Earth, so particularly with plants exposed to environmental stress like drought or pathogen attack and so on, and thought that the spaceflight example would be another great way to test as to whether we can pre-adapt plants to a stressful environment in the same way. This experiment requires 2 space flights because of that. And so the first flight will sort of set the epigenetic pattern within the seeds that are produced on orbit. And then those seeds come back to us. And then a comparison between the 2 sets of seeds, one that grew completely on the ground and the ground controls, and the seeds that grew on the space station will go back up together. And then an experiment will run again, and we'll take a look at the results of those 2 populations. And so comparison between the generations and between the seed sources, one with spaceflight in its generation history and one without. And we'll test whether the epigenetic transfer to the next generation helps the plants deal with that stress better than the first generation. That's pretty much it.
Thank you, Annalisa. We will now take questions for Annalisa. Please use *1 to enter into the queue or the hashtag AskNASA on social media. And while we're waiting, Annalisa, can you talk a little bit about how gene expression in spaceflight grown plants may vary between different types of plants?
Oh, sure. So gene expression is, you got to think of it in terms of the plant's toolbox. You know, how do you adapt to a stressful situation or any kind of situation? The patterns of gene expression reflect the tools that are used to cope. And so different plants have different strategies by which to cope with things. And so, for instance, if you were to look at the genes in obviously in a cactus plant versus the gene, growing in a marsh, you're going to have a lot of different strategies used to cope with those 2 different environments. And what we found in space is that the plants that grow in spaceflight environments, they turn on a lot of genes that are typically associated with stress responses. And so if we can understand better how to mitigate not only the stress of their environment, but also the kind of genes that are turned on
it'll help us grow better plants in exploration habitats or in orbital habitats.
Thank you. I see we have another question in the queue from Jim Siegel.
Thank you for taking my next question here. Maybe I missed it, but what sort of plants will the seeds be from? And for instance, are they the vegetables that are being grown on the ISS, or are these plants that might typically be part of some sort of exploration to the moon or wherever? Thank you.
Right. So the plants that we're using it's the model plant called Arabidopsis thaliana. And although it is the model plant used in a lot of different types of experimental, venues, both on orbit and on Earth, it is a member of the mustard family. And so it's really closely related to things like, mustard greens and collard greens and broccoli and a lot of the things that do comprise the crop venue for exploration habitats. And so a lot of the things that we are learning about how Arabidopsis responds to spaceflight is directly translatable to how similar family crop plants will also respond.
Thank you. We will now take additional questions for any of our speakers. Again, if you would like to ask a question, you can use *1 on your phone to enter the queue at any time. And while we are waiting for folks to enter the queue, I did have a question for Tony about the timescale with which the geological samples on IRIS will be exposed to the space environment, and what types of changes you expect to see on those samples as the length of time of exposure increases.
Yes, of course. Because these CubeSats are being launched from the ISS, they're quite low in altitude, so they will eventually re-enter the atmosphere and disintegrate, right? So we expect a complete mission duration of about a year and a half. And that's basically the exposure time once the commissioning of the spacecraft is complete, which we estimate about a month. And so what kind of effect do we expect to see? Well, obviously there's the UV radiation that will impact irradiation from the sun. There's also solar wind, change in temperature going from very hot to very cold. We also have vacuum exposure. So these type of effect will be monitored using these camera that will take picture of the sample. So it's kind of hard to predict right now what kind of effect we're going to see. I mean, if I was a geologist, I could probably elaborate a bit more, but I suspect what they intend to see is the intent is really to see if they can measure it in such a short time frame, and in the degradation within a year and a half, what it's going to look like.
Thank you. I see we now have a question in the queue from Jeff Faust of Space News.
Yeah, question for Kurt Costello. I wonder if you could speak in terms of the overall ISS utilization for science right now, how things are going, what the limiting factors have been, and also how things have gone in the last week while you had the Axiom 2 crew on the station and you had 11 people overall sharing all the station's science resources. Thanks.
Thanks, Jeff. First to the Axiom 2 question, we've had about 27 experiments run by the Axiom crew in conjunction with our ISS crew on board. It's been a very busy time up there. We are waiting for all the results to come back down when they land later tonight, and for them to be able to offload the samples and see how we did. But a lot of those were tech demos. This upcoming flight, SpaceX 28, is also a very good mix of biological payloads, tech demos, some Earth science monitoring, as you heard from the CSA team. And all in all, we've got about 31 investigations going up and being resupplied. Now, SpaceX 28 is still able to bring all that research, but it's also making up for the delays we had in our NG Cygnus vehicle arriving at station. So we're sending up lots of extra logistics, crew supplies for the crew to keep them going throughout the end of the year. So overall, we've been really exercising all the capability we have to do science on ISS. We are really looking forward to NG-19 later this summer, and then also SpaceX 29 at the end of this summer, bringing up a big collection of science that we've been accumulating on the ground ready to go.
Thank you, Kurt. Again, if you have a question, you can use *1 to enter the queue or hashtag AskNASA. And seeing no other questions, I would like to thank our speakers today and also thank the members of the media that tuned in, as well as all of our listeners for your interest in this important science, research, and technology that are launching aboard NASA's SpaceX CRS-28 mission. You can follow all the research being conducted during Expedition 69 by visiting nasa.gov/station research news or following ISS_research on Twitter. Stay tuned to nasa.gov for regular updates in advance of the upcoming launch. We are targeting June 2nd at about 4 p.m. Eastern Time for our pre-launch news teleconference, and then our launch broadcast coverage will begin at 12:15 p.m. Eastern Time on Saturday, June 3rd. As a reminder, you can listen to a replay of this teleconference by dialing 866-407-9261 or by visiting the NASA video channel on YouTube. Thank you all for joining us. This concludes today's telecon.
Thank you. You may disconnect at this time. Host, please stand by for your post.